Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension here at UW-Madison, and on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Peter Favreau. He works as a postdoc at the Morgridge Institute for Research. He was born in Jacksonville, Florida, and graduated from high school in Ocean Springs, Mississippi. And then he went to the University of South Alabama for his undergraduate degree in physics. He stayed at the University of South Alabama to get his MS and PhD. Both of those were in biophotonics. He came to Madison in 2016 to be a postdoc in Professor Scala's lab. He's here to talk with us about illuminating better cancer treatments with light. Now, many of you know that the motto of our fair university here at UW-Madison is Numen Lumen, and that Google translates directly as divine light. And I hope you will consider the divine as the verb, because I think what he's doing is divining how cells look and grow, and different cells at different stages of cancer look, and how we can use that to speed better cancer treatments. It's not every day I get to use a Latin phrase to introduce folks. It's going to be interesting to hear what he has to say about this brand of Numen Lumen. Please join me in welcoming Peter Favreau to Wednesday Night Lab.
All right, the first test. Can everybody hear me? All right, I passed the first test. So thank you so much for having me here tonight. I'm really excited to talk to you guys about some of the really exciting work that's going on just across the streets over at the Morgridge Institute for Research. Now, I have the distinct pleasure of actually having worked at the Morgridge Institute for Research now for a few years as a postdoctoral researcher. And it's a beautiful building, as you can see here. But in addition to just being a beautiful building, it has an incredible set of researchers that work there on some really exciting science that's going on. If you take a look inside, there's always something going on inside of Morgridge, probably something to do with the community. There's constantly some kind of outreach event, some scientific opportunity. Every time I walk in the morning, there's always some new sign that says I need to walk all the way around the building because it's currently blocked off because of this new conference that's going on. So there's always something going on. It's always something very exciting. But one of the best parts about it is this philosophy that Morgridge goes by, which is practicing what's called fearless science. And really what that means is that the researchers there are encouraged to pursue ideas that are potentially very difficult to maybe get funding for, because there's a high, they may not necessarily be the easiest experiments to follow through on. So the whole idea here is to practice really cutting edge work, and there's all sorts of amazing researchers I get to work with on a daily basis. Now, the one that I work in, the laboratory that I work in, is the Optical Microscopy and Medicine Laboratory, and the principal investigator there is Dr. Melissa Scala. This picture is actually pretty interesting because when I first came here in 2016, there were like 3 or 4 of us. And as you can see, there's a few more than 3 or 4 now. So the lab continues to grow pretty much every month. It seems like we're talking about someone else. So it's really exciting because we have all these new ideas and collaborations that we're doing. And some of that I'm going to talk to you guys about tonight. So with that, what does optical microscopy in medicine actually mean? What am I talking about? Well, to put it kind of simply, we're talking about light using microscopes, microscopy. This is a very expensive microscope that I'll get into in a second that I use fairly often. It's called the Multiphoton Microscope. And we use this to better treat patients, to improve patient outcomes, and especially in cancer. And we want to basically improve precision in medicine. Now, what do I mean by precision? Well, there's this paper that came out where they modeled a cancer growth. As you can see here, this is a model of a tumor being grown, where different cell populations are colored differently. They gave it a drug, and what they found is if they modeled this cancer development, if there are some cells that are resistant to those drugs, the whole cancer will grow back again. And we've probably all heard about this before, right? You go immediately in for some kind of cancer therapy. Initially, your cancer goes into remission, and then unfortunately, later on, it might come back. And this is because there are certain subpopulations of cells that are present in cancer that may be resistant to the drugs that we give them. So what we need then, or to put this in another way, currently, one of the most common methods of actually combating cancer is to use chemotherapy. And chemotherapy kills both cancer cells and healthy cells, so it's sort of like a sledgehammer when it comes to curing cancer. Now, the perfect drug, though, doesn't hurt your healthy cells, of course, it just hurts the cancer cells. And what I just described a second ago was most drugs that we give are going to end up potentially leaving behind resistant cells as well. So this is sort of the problem when it comes to treating cancer in patients, and it's a very patient-specific problem. We have to find more precise treatments to help these patients. So the challenges going forward, and what our lab is particularly interested in, drug identification for cancers is costly and time-consuming. So a patient comes in, they may have a standard set of drugs that they can get, but ultimately it can take time and it can be expensive to figure out exactly what the right drug cocktail might be for that patient. And there's really no intuitive method for doing this at the outset. We're getting better about this, but we need some intuitive way of matching drugs, and not just drugs that are currently the standard of care drugs, but potentially more targeted therapies that might offer even better results than the drugs that we currently know about. And finally, I mentioned this just a second ago, but these drug-resistant cell populations that remain after treatment, we have to find some way of destroying not just the cancer cells that are immediately going to be affected by the treatment we give them, but all the cells, while also, of course, not hurting the cells that matter, the healthier cells. So these are the challenges then that really guide a lot of the work that goes on in the optical microscopy and medicine lab, and especially guides a lot of the work that I do. Now, how do we address them? Yeah, that's really what I'm going to be talking about with you guys today. There's sort of a three-pronged offensive that we undergo, one of which is to use patient-derived samples, and I'll kind of get into that in a second. We also use a contrast technique called fluorescence. And finally, we use something called single-cell analysis, and I'll describe what all of these mean eventually. But first, let's just start with patient-derived samples. What do I mean by that? Well, traditionally, if a patient comes in and they have a tumor or some kind of cancer, a physician might give them a set of drugs or have options available for that patient. And if they don't know exactly what to give that patient, they may have to kind of go through one at a time and figure out and see if the cancer itself shrinks, and then they know it's getting better. Of course, this can take time, and this is, as I said, is one of the problems with finding a more precise treatment for these patients. Now, a better way, a potentially better way here, is patient-derived tumors in a dish. And I'm just going to call them organoids from now on because tumors in a dish is kind of a mouthful to say constantly. So what does that mean? Well, instead of trying all these drugs on a patient one at a time, we take parts of that patient's tumor out and we regrow them in dishes. And now, whenever we want to try these drugs out, we can actually try them on that patient's individual tumors, basically. And instead of hurting the patient or sending them through this, we can get fairly quick results about whether or not this drug is going to work one way or the other. This is just a couple example images of what these guys look like. And one thing just to take away from this is that they vary pretty considerably. These are from two different patients. So, the specific tumor and how they look are going to vary depending on the patient that comes. And that kind of gets to the whole heart of one of these questions, which is every patient's different, so they all require a different set of drugs to treat them. So treatment response is generally measured in these organoids by measuring a reduction in organoid diameter. So similar to how you would measure whether or not a treatment is working in the human body by measuring, let's say, a CT or X-ray, a tumor shrinking, in the case of organoids, you would just wait and see if the organoids shrink. That's a very common way of doing that. So before treatment, you take some kind of measurement about their diameter, and then after treatment, you'd make that same measurement. And you would assess whether or not the organoid itself is shrinking. If it is, then you're hopefully on the right track. Now, one of the issues here is sometimes you get organoids that look like this, which, I don't know if you're me, but I don't know where to draw the line to figure out where the diameter is. And this is actually more common than you might think, and so it's one of these kind of confounding problems if we're going to be talking about doing diameter reduction measurements in organoids. We have to find a better way of assessing whether or not a treatment is working that isn't so dependent on them being spheres or circles that we can measure the diameter. So basically, is there an easier way to measure treatment response? And I'm here to tell you, yes, there is. And that way in our lab that we use is fluorescence. So what is fluorescence? Well, fluorescence actually has its origins over 150 years ago from this guy. He's a very famous guy, George G. Stokes. In physics, he did all sorts of amazing equations that I had to memorize, and I sort of hate him. But I also really admire all the different experimentation that he did. One of the experiments he was working with, he was sitting in a lab, and he was working with, back in the day, a lot of these researchers would basically just have minerals laying around that they had because they were doing all sorts of fun experiments with them. And one in particular, fluorspar, he had laying around, and he determined that under a certain color of light, wavelength of light, it would glow blue. And this is a phenomenon that had been witnessed by several researchers at the time, but no one really knew what the deal is. How is this happening? Why is it happening? And George G. Stokes is responsible for figuring out what's going on and then coining the term from the mineral that he noticed this in. He called it fluorescence. So not all molecules actually fluoresce, of course. It's just given the right conditions and the right chemical structures, some of these molecules will. And generally, it's a matter of how the organic structure of these molecules is and whether or not they have free energy available to actually fluoresce. So to go a little bit more into detail about that, if we consider you have these orbitals, and electrons typically have energy states that they can move up in, depending on whether or not they're excited with some form of energy or light. So if we call this very bottom energy state the ground state, S0, and this one over here at a little top, this is the excited state of an electron. If you have an electron and it's excited by light, a certain wavelength of light, depending on the structure of that molecule, the electron will jump up into the excited state. It can't hang out there forever. At some point, it's going to come back down, and in the process, it'll emit a photon. And that emission of light is fluorescence. That's what when I'm going to be talking to you guys about later, is what we are basically measuring, is this emission of a photon. So as I said, fluorescence has been around since around 1856 in terms of our understanding of it, but since then it's sort of exploded into a lot of different applications. In 1871, that's when fluorescein was invented. That's the first synthetic fluorescent molecule. If any of you guys have been to Chicago during St. Patrick's Day, they color the river green. I didn't know this, but they just threw a bunch of fluorescein in there. I don't think they actually use fluorescein anymore. It's probably toxic or something. Yes, there we go. Very toxic. So they do something else, but it used to be fluorescein is what they used. A few years later, they invented some method of actually being able to look at microscopic organisms using and fluorescence, and they invented the first fluorescence microscope in 1904. And you can see it's a very crude microscope design compared to what I showed you a second ago. But that's when the first fluorescence microscope was invented. About 40 years later, immunofluorescence came about. And immunofluorescence uses a combination of antibodies that are specifically tagged to proteins of interest to not very specifically find contrast of those and identify them in living organisms. Now, just a few years ago, we finally found a way of manipulating the genetic code of organisms, especially jellyfish is where we first got this. But basically, the researcher who found this found jellyfish in Seattle, right, or Vancouver, and he identified the protein that was causing these jellyfish to glow, and he ended up being able to put that into other organisms. Here, in this image, you're actually seeing bacteria that have been genetically encoded with different variants, so what's called green fluorescent protein. Of course, they're not all green here. Some of them are red, because depending on how you modify the structure, you can make different colors fluoresce. So this is sort of like a very bare bones history of fluorescence. And just to tell you guys, really, that fluorescence gives us a lot of contrast to help us find answers in biology and medicine. Now, that said, can fluorescence help us find the best cancer drug? I've given you guys this introduction. Now let's bring it all back to medicine. Well, in the 1950s, there was a researcher who was very interested in fluorescence in your cells, and that's everyone in here. If you had the right wavelength of light on your cells, they would fluoresce. And he was looking into metabolism of cells. And this guy, Britton Chance, who's actually a very fascinating character, I think he was an Olympian gold medalist in sailing. And I think he also, I don't want to speak too high, but he did a bunch of really exciting research in cellular metabolism. He identified some naturally fluorescent molecules in cells. And basically, he was looking at mitochondria. And what he found, if you look at the processes that are going on, metabolic processes around the cell, in the cytoplasm, there's glycolysis that produces energy. And of course, in the mitochondria, we have oxidative phosphorylation. And when he was doing these experiments, he was finding that there are two molecules in particular that with the right wavelength of light, they would glow, they would fluoresce back. So he did a lot of experiments showing that he could actually manipulate the intensity of the light from these fluorophores, these fluorescent molecules, and showing that they're actually from NADH and FAD. And he also showed that depending on how these cells are metabolizing, this fluorescence will change, which is, of course, a really fascinating and strong contrast method, because now I don't have to put something into a cell or genetically encode something. I can just use the right wavelength of light and look at how metabolism changes, how things grow in cells using these two fluorescent molecules. Now, another part of this is, so we have these two molecules where NADH fluoresces predominantly in the cytoplasm and FAD fluoresces predominantly in the mitochondria. And naturally, you can take a ratio of the two, and it kind of gives you like a ballpark way of estimating how these cells are metabolizing. So we now call this the optical redox ratio, but there's actually a few different variants of how to calculate this. But basically, by taking the fluorescence of the NADH divided by the fluorescence of FAD, you get, like I said, this kind of map of electron donors and acceptors that are present in the cell, and really just how the cell is metabolizing. Our work has basically shown that this ratio, depending on if it's lower or higher from an untreated sample in cancer, if it increases, the optical redox ratio increases, then we typically say that cell is non-responsive to treatment. Whereas if it reduces, then it's actually responsive to treatment. And I'll get into a little bit more about what this means, but I just wanted to prime you guys with what these are going to mean. So the optical redox ratio, what does this actually look like in space? Well, this is an image of just some cells that were acquired with the right excitation wavelength and emission wavelengths to see NADH fluorescence in cells. And what's interesting here is these dark spots are nuclei of these cells, and around that, where all the fluorescence is happening, is the cytoplasm. And of course, as I said, NADH is predominantly fluorescing in the cytoplasm of cells. So you get this very specific contrast, spatial contrast for these different fluorophores. And additionally, with the right excitation wavelength, we can look at FAD fluorescence as well. And if we were to combine these two images together, we can get this, essentially this map of how the interplay between these two molecules, the intensity of light. So if you're looking at this, the brighter, more orange areas, there's typically more NADH that's fluorescing. And conversely, if it's a little darker or more towards the purple, there's generally more FAD that's fluorescing in that region as well. Now, how do we actually acquire those images? Well, as I said, we use a very sophisticated microscope. It's a little more sophisticated than the one 1904. This is a multiphoton microscope, and this is predominantly what we use. Why we use a multiphoton, there's several reasons why we use it. Typically, for multiphoton microscopy, I know I said multiphoton, but it's helpful to compare it to what many researchers use right now, which is a confocal microscope. Confocal is also very excellent at exciting these molecules and looking at different cell structures. But by nature of using the confocal, you get a lot more dispersion of this light at the focal plane. And that focal plane is kind of, let me see if I can get this mouse, right here. So yes, you'll excite whatever you want to see, whatever molecules right here, but you also get all this called out of focus exposure of this light, which all that light is going to interfere with the signal that you want to see. Now, you can generally improve that with confocal, but multiphoton, by its very nature, is able to reject all of that light that's outside of it. So that means that you only see where that little point of light is right here. So if I'm scanning a sample, I can very specifically excite things instead of getting out of focus excitement of different fluorescent molecules. So this is very powerful because of that, the specificity and rejection of the out of focus light that you get. But it also, and I'll get into this in just a second, it allows you to basically do very three-dimensional imaging by its very nature. So just to kind of take you guys through a really quick study, we're going to be talking about the using fluorescence in organoids, but I want to show you what that looks like if I were to use fluorescence in organoids in kind of a trial study where you're interested in determining whether these targeted therapies could be used in colorectal cancer patients. So colorectal cancer, this is an area of research that I've spent a lot of my postdoctoral research working on. Unfortunately, colorectal cancer is a fairly prevalent cancer. There were 130,000 new cases in the United States alone, and then there were 45,000 new deaths as well. It's a fairly prominent cancer, and right now there's a 5-year survival rate of 65%. So of those patients who survive, or all the patients are going to receive some kind of, potentially could receive some kind of therapy to treat their disease. And that therapy may be effective, but they also have to go through the process of actually dealing with potentially ineffective therapies. So some of these patients are going to receive a lot of toxicity from drugs that may not be as effective, and they have to go through multiple rounds of chemo to eventually eradicate their disease. So there's a need to, as I said before, find a more precise method of determining the best drug for these colorectal cancer patients. So what I'm going to show you is organoids from 2 different patients, and we always have an untreated control, and then we compare that to one of these targeted therapies called MLN-0128. The numbers don't matter. It's one of the targeted therapies that targets cancer metabolism, and so does this other drug, ABT-263. Collectively, we're interested in seeing how each of those drugs performed on the patient's organoids by themselves, and then we're also interested in how they would, as a combination therapy, be effective, which gets to one of these things is that we can basically try all sorts of combinations of drugs because we're just testing it on this patient's tumor outside of their body. So it allows us to do all sorts of drug screening techniques that we couldn't have otherwise done. So we'll regrow each of these patients' tumors in dishes, and then using this microscope, we can image them for the fluorescence of NADH and FAD, and we can collect the optical redox ratio images from each of these. So as I said, the untreated sample here, this is not treated with any kind of drug. This is just to show you what it compares to when we actually treat it with something. And when I look at this, what's immediately apparent to me is just how different in shape they kind of look. You have smaller organoids in the top patient that are kind of clumped together compared to the bottom patient's organoids, which is just kind of one big organoid that's more spherical in nature. And so the organization of both of these patients is very different. So definitely on just like a qualitative way, you can see there's a difference between these organoids. And then when we gave the treatment to these organoids, what you see is a dramatic difference also in how they respond to treatment. And in fact, the top right one basically completely eradicated a lot of the outside organoids that are present, leaving only a few organoids remaining. And the bottom one didn't, I'll show you in a second what actually happened. But basically, this combination therapy was also revealed quite a few interesting features about the 2 organoid patients. Now, can we quantify differences between the treatments themselves? Well, what I'm going to show you guys is basically plots of how this looks if after we treat them. So here are the untreated optical redox ratio results. Now, anything that's above this line is going to be something that's not effective. It's non-responsive to treatment. Whereas anything below this line, this untreated line, is going to be effective at treating this cancer. So here you can see that all of the drugs actually were below this line, but especially this combination therapy was extremely effective. The optical redox ratio was very much reduced compared to the control. So we would call this a responsive organoid compared to, unfortunately, in this patient's organoids where they saw basically no effect on this drug. So unfortunately, this patient was non-responsive to these therapies, or at least the organoids were non-responsive to these therapies. So as we can quantify this, we can get information about how the patient is metabolic, or the patient's organoids are metabolically responding to these drug treatments. And we can do this across a wide variety of different treatments. But can we actually push this fluorescence contrast further? Can we go even deeper with the data and ask more questions about what's going on metabolically? Well, I talked to you guys about what fluorescence is in general, but there's another element of contrast that we can actually get. If we were to time the point at which this electron is excited by light to the point at which it gets back down to its ground state, this is something that's called the fluorescence lifetime. And this is a very interesting contrast method because the fluorescence lifetime is very specific for each fluorescent molecule. NADH and FAD have their own fluorescent lifetimes compared to each other that are distinguishable. They're also concentration independent, so it doesn't matter how much NADH is present, they have the lifetime is consistent. As I said, they're molecular specific, but they also change, so the lifetime exists on a continuum, and I'll explain what that means here. NADH, if it's bound to a protein, its lifetime will actually get longer, whereas if NADH is free, it has a shorter lifetime. And so this phenomenon, if it's bound to something, it has a longer lifetime. If it's free, the lifetimes are shorter. This can help us distinguish NADH versus FAD and also tell us whether or not what the local environment consists of for NADH and FAD. Now, how do we actually measure fluorescence lifetime? Well, in our lab, we use time-correlated single photon counting, which is really a lot of words. It's very kind of hard to say. But what is this? What is time-correlated single photon counting? Well, I'm going to do my best to explain it. Basically, if we were to graph the counts versus time, and I'm just going to bring up this energy states again, we have this laser that pulses our sample, and we have this initial excitation pulse that will excite the molecule or the electron up to an excited state. It'll have a characteristic emission where a photon is emitted. And that photon is detected by, in our case, we use photomultiplier tubes, so you can use cameras as well. But basically, we can use this to time the time it takes for it to be excited and then subsequently relax back down to its ground state. Now, not every electron is going to experience the same time. They'll come in at slightly different time points depending on basically quantum effects. So understanding how this works, these photons, and how they're actually received, I usually think of Connect 4 because it's basically how we're going to end up building a histogram out of these photon counts. So if you think of each one of these columns as basically a bin where photons have a certain time it takes for them to get to the detector, some photons are going to come in at this time, others are going to come in later. And basically, over the course of about a minute, you're going to get several different time points that you can use or that build up. And you can actually fit something to this data, an equation to this data, and use that to inform you about the fluorescence lifetime characteristics of your images or your sample. Now, we do this on a per pixel basis. And what this basically means is that after we go through each one of these pixels, we can create an image and use that. As I said, I just showed you guys kind of the same image, but we can get the fluorescence lifetime on a per pixel basis and use that to do even more quantification of our samples. Now, how do we measure the lifetimes from this histogram? As I said, we can actually fit a bi-exponential fit to this data and understand how it's binding or how NADH and FAD are actually binding in this case. And in the process, if I false color this kind of shows us what that looks like. So something that's more blue in this image is more free NADH. Conversely, something that's more orange or red is going to be more bound NADH. And as I said, this exists on a continuum. So there's going to be some, depending on the binding partners present, that in each one of these cells, you're going to have a slightly different lifetime for the NADH. So this is very interesting. I'm showing you guys 2D images of basically a spatial map of the fluorescence lifetime for each one of these cells. But I mentioned earlier that the Multiphoton Microscope is great because it can actually go through a whole 3D sample, which is really fascinating. But how do we actually do this? How do we incorporate this then into our organoid imaging, which are 3D samples, right? Well, using our multiphoton microscope, we can take these fluorescent lifetime images one at a time through our sample. And these images are actually about 12 microns in depth difference. So basically, we take an image, and then 12 microns above that, we take another image, 12 microns above that, we take another image. And then if we want to combine these images into a single 3D image, we get something that looks like this, which I think is just a really cool and interesting way of visualizing these 3D structures. Microscopy, if you don't use this technique, if you were to just look at the diameter, remember how I originally looked at the diameter measurements, and we were looking at this kind of crazy shape and asking, where can we actually draw the line for diameter? Well, if we have the other problem with that is that there's this whole, it's kind of like an iceberg problem, that there's the underside of that organoid might have its own shape as well, that we can't really see. So we need to use some method like a multiphoton microscope to actually see the entirety of the organoid. Using this allows us to actually visualize the entirety of the organoid. So then with the additional, if we add the additional contrast from Fluorescence Lifetime, you can kind of get these really beautiful images of both NADH in this case, but we can also do FAD. And what I want to draw your attention to is that they actually excite two very different parts. So you're getting two different kind of contrasts from NADH and FAD. What's really interesting in the FAD image, actually, unfortunately, looks like it stopped spinning. But as it was spinning, you might have noticed that there was sort of a blue core in some of these. And what that is the cells that aren't receiving enough nutrients will actually start to collect in the center of these organoids, and they'll die. And we can see that with using the FAD contrast better than we can see it with the NADH contrast. So you can see differences in metabolism at a glance by using this kind of approach. So then the challenges, the first two challenges I think we kind of address using organoids and fluorescence, namely that we can use those techniques to find better ways of identifying drugs, and we can also find better ways of matching those drugs to patients. But I haven't really spoken to this last point, which is finding a way to identify these drug-resistant cell populations that may exist, even after a patient undergoes some kind of treatment. So the typical way of taking an image like this is just another one of these fluorescence images. This is NADH fluorescence from an organoid. You can take the average intensity across this entire image and get some kind of a quantitative readout of what you're looking at. But that doesn't help you identify the individual cells and how they're responding to treatment, which is really the question, right? How are each of the cells that comprise that organoid, how are they responding to the therapies that we're trying out? So that's what we want to know. So our solution is to use single-cell analysis, is to go deeper into these images and try to tease out information on a per-cell basis, and not just look at the whole volume of an organoid or just one slice. We want to look at the individual cells that are in each one of those images. So how this works, we use some software that allows us to take an input image like this and then segment the nuclei that are present there. So the red regions here are all nuclei. And then if we extrapolate out a few pixels, we can find an outline for each one of these cells. And then, of course, the difference between where the cell outline is and the nuclear outline is everything in between, which is the cytoplasm. And, of course, in the cytoplasm is the mitochondria and the cytoplasm where the NADH is present as well. So underneath where this region is, we can tease out all this fluorescent information. And now we're not talking about how does this whole image respond to treatment, how does the organoid that's present here, we're talking about how does this individual cell respond to treatment. And that gives us, the data that we get from that gives us population data, populations of cells that are present in each one of these images. So I'm going to bring you guys back to this Connect 4 analogy, because I think it works well here too. But how can we actually interpret these populations of cells, the results from that? So if we again divide this up into columns, the optical redox ratio, if we had, each one of these cells had a corresponding optical redox ratio measurement, 1 through 7, say, some of these cells are going to kind of differ slightly in their metabolism, so they're going to have different optical redox ratio measurements. And if we were to look at this, we could actually fit some line over this. And what you're seeing here is basically two populations of cells that kind of center over either 2 or 5. It's just an example, but this is really what we see when we look at the data. And so if we have these two populations of cells that are present in a single image, we would call this a heterogeneous response. And what that I'm really saying here is that some of these cells are going to have a higher redox ratio, and some are going to have a lower redox ratio depending on the treatment that they get. So what we would really want to see, if you're a patient and you're looking at the individual cells that are present in an organoid, you would want to see something that looks like this. We just have one single population of cells that respond the same to the drug. Because if there's a population of cells that's responding differently than the other one, that means they might be resistant to that therapy, and you'd have to find some other targeted therapy to take them out. And so we would call this a homogeneous response to the drug therapy we're talking about. Now, what does this actually all look like in patients? So everything I've told you up to this point, we've done a considerable amount of experimentation and looking at, we get patient samples in and we make organoids and then we try several different therapies. But what does this whole process look like if a patient walks into the doctor's office and we run this entire process? Well, there was a study that actually just got accepted into the Clinical Cancer Research Journal where we attempted to prospectively identify the best treatment for a patient with metastatic colorectal cancer. So this patient, there was a surgical sample we received and we tried a few different standard of care therapies. So 5-fluorouracil is a standard of care therapy, 5-FU, for colorectal cancer, as is oxaliplatin. And we looked at the combination of the two, and we tried these drugs on this patient's organoids to see whether or not that patient would respond, or the patient's organoids would respond. And so these are just some representative images that we got. So this is the untreated one. This is not treated with any kind of drug. And we looked at the lifetime as well as this optical redox ratio. And we compared this across all these different treatments. And I think the thing that jumps out to me immediately as I was looking at this is the Folfox, the fluorescent 5-FU plus the oxaliplatin. And what you can see, especially in the NADH lifetime, is that you have far more bound NADH, and you have these these blue cells that are surrounding it. And basically what's happening is that these organoids are dying as a result of using this kind of treatment. So we were very excited about this, but we can't just give a patient this based on what we saw. We wanted to compare it to another set of data. Now, before I jump into that actually, so we took the data, the quantitative results we got from that, and as I showed you guys earlier, we wanted to look at the populations of cells that were present in here. So what you're seeing here in white is the untreated control. And then what you're seeing in red is the combination therapy that we gave, the 5-FU plus the oxaliplatin. And so this untreated, as I said, we're only seeing one of these Gaussian curves, one of these distributions, and we call that homogeneous. But then with the treated, the 5-FU plus oxaliplatin, we get this heterogeneous response. We have this one subpopulation that's on sort of the lower right that didn't respond as well to the treatment. And then we have this large peak this group of cells that did respond to treatment. So even though this is heterogeneous, this is actually great because the patient the organoids were responding in some form to treatment. And we wanted to confirm that this was also happening based on diameter measurements, which is sort of the gold standard for looking at organoid treatment response at the time. So we compared this to the change in diameter, and what we saw is this exact same peak present in theirs too. They had this response that they saw, so we confirmed our results from the change in diameter that we got from our collaborators. And this patient, based on this data, was given this combination therapy. And what I'm going to show you here is just this CEA marker, which is just a common marker for cancer. And basically they did a CT scan before the drug was given, and what that red arrow is pointing to is their tumor. And then after being given the therapy, they started to see this marker of cancer just drop. And in fact, when they did a follow-up after 2 months, they saw that the tumor had dramatically shrunk after being given the treatments that our organoids had prospectively said were going to help this patient. So this is really exciting work because it shows that we can implement this in a patient scenario. Now, of course, there's a long way to go before we could actually use this commonly, but these are the sorts of experiments that we're really excited to kind of jump into and find a way to do more of. So just to kind of wrap this up, as I said, our lab is very interested in doing these kinds of experiments and finding ways to improve precision in medicine. I showed you colorectal cancer, but we are actually interested in a lot of different cancer types, not just colorectal cancer. We're looking at pancreatic cancer, which has just a devastating mortality rate. And there are just so few options for patients who have pancreatic cancer. There desperately needs to be a way of helping those patients find the best drug to treat their disease. Same with all of these, breast cancer, oral cancer, neuroendocrine, and we're constantly trying to find new approaches because this approach of using fluorescence and organoids can extend to more than just colorectal cancer. It can extend to all these and others. So we're excited to continue to reach out for collaborations at Morgridge and beyond in UW-Madison. And with that, I just want to thank the people in my lab, especially my PI, Dr. Melissa Scala, and several of the members in my lab who helped make this. Definitely the Deming lab, Dusty Deming over at the Colorectal Cancer Institute did all of this patient collection, or patient sample collection, as well as a lot of the organoid training and growth. And especially thank you to all the patients that consented and allowed us to do these kinds of experiments. So with that, thank you so much. I appreciate your time and your attention. And if there's time for questions, I'd love to answer any.
Yes. When you were talking about how some cancer cells are actually drug resistant, it of course made me think about how certain bacteria can develop drug resistance, and so I was wondering if there's any reason to think that the standard cancer treatments might be making cancers hardier across the population. But having said that, I know that some cancers in recent years have been linked to a virus, not a bacteria. So then I was thinking if maybe my thinking on this was a little bit off in comparing it to bacteria. But I guess I know that's kind of a two-part question, but I wanted to see if you had any thoughts on that, and I know it wasn't exactly the topic of your presentation, but it made me think of it while you were talking.
Yes, I think that's a great question. Let me make sure I got it. So you're asking whether or not how kind of drug treatment resistant occurs in these cancers and how that might relate to bacteria and viruses. Is that true?
Sort of. I guess I was wondering if it's if it's a good way to think of cancer cells as being like bacteria insofar as they can be drug resistant, and insofar as that is a plausible analogy, if we then can, I guess, wonder whether the standard cancer treatments are making cancers hardier in the population.
Yeah. So are cancer drugs making cancers themselves hardier, the cells present that are hardier? And unfortunately, I think the answer is yes. Basically, you can sometimes have the case where cells will get sensitized to a drug, and then they'll just keep going, and the disease itself will come back. So that does occur, I think. And one of the things about this is one of the interesting areas of research is trying to find a way to better predict which cells are going to sort of come back after the drug has been given. And there's definitely studies that are going on right now. We're very interested in that too, because I think with this kind of contrast technique, if we can see it in an organoid, you could definitely tease out those cells individually, potentially regrow them as organoids, and then do more experiments on those and figure out how the metabolism shifts depending on the drug treatment you're looking at. It's a really interesting question. I think cancer itself is one of these things that is, of course, a multifaceted disease, and there's no easy solution. And one of the hardest parts is dealing with the fact that the cancer can come back even if you've basically annihilated most of the cancer at the outset. Yeah.
I guess following up on that, is the resistance from the fact that that particular cancer cell then replicates at some point, and there's an evolutionary process by which the daughter cells are more, by definition, more resistant? Or are we talking about a cancer cell that just a single cancer cell that just is resistant itself? I mean, is there a cellular replication that's occurring that causes it to come back?
So yeah, if your question is whether or not there's some intrinsic part of a cell that either makes it resistant, or is there some kind of post-mutation that causes the cell to be resistant? And I'm not entirely sure. I think that there are cells that, one way of immediately assessing whether or not drugs will work on a cancer is to look at their driver mutations. So do some kind of next level genetic sequencing, look at a patient's tumor and establish what are the mutations that are present in that patient's tumor. That's true for cells. So if the disease is especially heterogeneous, some of those cells may intrinsically already be resistant to therapy. And we kind of already see this in the organoid data that we look at. It's also potentially the case, I think, that if you sensitize some of these cells to a drug and it doesn't outright kill them, as long as they persist through the treatment, they could potentially gain some kind of protection from that drug and also continue to replicate. So it may be a combination of both, although I'm not 100% sure. Yeah?
In your discussion, you talked about photomultiplier tubes and cameras. Do you have a camera that has a number of photomultiplier tubes, or how did you derive that three-dimensional image with photomultiplier tubes and counting?
Yeah, so that was taken with a photomultiplier tube, and basically we went at a per pixel way. We just collected the photons at each one of the pixels, and we rasterized across the sample to create our image. And then we moved our stage up a little bit, took another image, and then we did that several more times, until we got this full volume. And then we were able to reconstruct the entire volume of our sample and give you that image that you just saw. So it's a process of taking one image, moving the stage, taking another image, and so on. Yeah?
With precise targeting, is the goal to annihilate all of the cancer in a particular patient? But that's not successful yet.
Yeah, so your question is the goal to get rid of all the cancer at the outset? Yes, I think that the goal, of course, is to do that. Now, whether or not that's possible for every cancer patient, we don't have all the most effective cancer drugs to say that this definitely will always be the case. That said, I think that one of the strengths here is that you can try all sorts of new therapies that maybe you wouldn't at the outset do. But with this kind of technique and approach, you can be like, okay, I've got this drug over here that works based on these driver mutations in this cancer. Will it work in this cancer? And you can try it in combination with other drugs and see if before you didn't know this was going to work, if it now works with this particular cancer. So I think that this allows us a lot more customization, so to speak, of how we treat patients' cancers.
So there should be less developing of resistance then, if you can get rid of the cancer.
Right, yes. That's another plus, I think, here, is that if you have better targeted therapies, then you don't have to deal as much with potentially these remissions that occur. Yeah. Yeah.
How much time does this analysis take now? And I guess what would be the rate limiting step of this? Would be growing the organoid? Yeah. Or would it be the analysis itself?
Oh, that's a great question. Right now, I think it's growing the organoids. It doesn't, that said, it takes probably, you can get a response from this. I think the quickest we do it is like a week. So, which is a much faster turnaround than, say, waiting for a tumor to shrink within a month and then having to CT scan or do another imaging to verify that and have the patient come in. So we have a quick turnaround on that. And basically, when you get these organoids growing, you can do several passages and continue to look and test new drugs as well. So you're not kind of waiting. The patient doesn't necessarily have to wait as long as they would have otherwise to do that. But right now, it's definitely waiting for the organoids. The analysis, there's another element to this, which is automating the process to make it even quicker, which is something that's going on in our lab as well. We do all sorts of we're trying all sorts of methods to streamline the process of looking at the analysis so it's quicker, and also incorporating things like machine learning so that we don't actually have to be, we can train a computer to do a lot of the segmentation and then give us this kind of information back and be able to recognize this organoid is responsive versus this one is not. So those things are very exciting, and we're very interested to see how that's going to play out from here on out. Yes?
If this is precision for a particular patient, does this help you determine the dose and the duration of the treatment for that patient?
Yeah, so your question is if this kind of precision method is going to help us find the right dose for that patient and the amount of time that patient ought to be on that drug. And I think the answer is yes, definitely to the first, because we can try several different dosages at once on the patient's organoid and see which one of those dosages is the most effective without annihilating all the other, potentially other cells that are present. The other point about how long that patient should be on, I think that's going to end up being more of a, the physician's going to have to assess that probably on a per patient basis more. I could see it being the case that if you're able to do more longitudinal studies over time, you might be able to tease out, okay, this organoid was responsive to treatment for X amount of days, and then we saw that it became resistant or something to that effect. And that might give you some information about whether or not, how much time that patient would actually need to be on the drug. Yes?
The redox ratio we mentioned for the multiple cells looks like it was a ratio of 4 or 5 for number of cells that responded to the treatment. I was looking at your graph, and does that redox ratio vary in terms of efficacy from patient to patient? And also, if a patient has a relapse in terms of the cancer coming back, do you then make your analysis and try a new drug or say the same cocktail?
Yeah, so the first question you're asking whether or not the redox ratio varies from patient to patient, is that?
Yes.
And the answer to that is yes, which is why we always compare it to that patient's untreated control. So we compare how effective a treatment is based on how much it differs from this untreated control. And I'm sorry, what was your second question?
Second question was, if the patient relapses and the cancer seems to have come back, do you then try a new drug or a cocktail of drugs? You sort of add the new drug?
Sure, I think it could potentially be an iterative process, but the hope is that you wouldn't have to do that at all, right? Is that at the outset you'd find some kind of, you'd find the most targeted therapy that would kill the cancer immediately. That said, if it was the case that if their cancer came back, you would definitely be able to still go through and do the process again and identify a new drug. And I could foresee it being the case that you would have sort of a suite of drugs or a cocktail of drugs that you knew were effective now, and maybe you try those again if the remission occurred. So it's an iterative process, and it isn't one of those things that you'd have to just do once and you couldn't do it again. Yeah. Oh.
When the organoids are growing, are they stuck to the plate permanently, like a bacterial colony?
That's a really great question. Are the organoids adhering to the plates? And we actually put them in a 3D matrigel environment so that they grow into these 3D cultures. So what ends up happening is we kind of combine them with the matrigel and we put them on dishes, and they are suspended in this matrigel culture, and we put media over top of that.
So as soon as the pattern is established, that where they are located are like fixed stars in the firmament.
Basically, yeah. They don't really move around a whole lot, at least not within the time at which we take an image and we wait and see how the treatment responds.
So when you take the image, are you taking the image of the whole dish and being able to analyze several hundred organoids at once?
That's a great question. So there are, the images I showed there, that's not the case. Usually we go around and we take images of the organoids we see. If you have a lower magnification objective, you can take larger fields of view of the entire dish and do that with just, say, an epifluorescence microscope, so a microscope you might have in the lab that it's lower magnification. There's also a method that I've been working on very recently that's uses light sheet microscopy. And make a long story short, it allows us to look at entire volumes of hundreds of organoids if we want. It just, we're limited more by the size of the matrigel and the tube that we use to actually culture these organoids in.
We've had talks from astronomers about how they can look at a field of view of the sky of stars and look for changes over time. And it seems if that's what you're able to do with this, you'd be able to, it's still screening and not selecting, but screening hundreds at a time with one shot from time 0 to time 1 to time 5 or whatever you're doing.
Oh yeah, no, I think that's pretty much where we want to go in our lab. We want to increase throughput, and we want to, because I'm talking to you guys about cell populations, which I think is incredibly important for understanding heterogeneity of response. But another question is, if we have hundreds of organoids in one of these wells, how does the population of organoids change, like over time with treatment? That's also a very important question, and that's something that our lab is moving into. And to do that, we have to find these quick imaging techniques and methodologies so that we can do that. It's a great question. Yeah.
How unique are these microscopy techniques? Is this something that's happening in a lot of labs around the country and around the world, or is this fairly unique?
Our approach is fairly unique. And multiphoton microscopy is, I don't want to call it a common technique, it's an expensive microscope. I think they're about $1 million. Henry, are they $1 million? They're very expensive. And as a result, that is sort of a limiting step in how many labs can use them. And our approach of using these organoids to screen using the multiphoton microscope is novel. That's something that is pretty much only done in our lab and maybe a couple others. Yes?
How do you choose your patients? Are they referred to you from UW doctors? And are they people who have not responded to other treatments? That's
a great question. So how do we choose our patients? And actually, the Deming lab, what I acknowledged there, they are doctors. And when they have their rounds, they'll go and they'll try to consent patients. And so really the answer is the patients come to us because they come in for their checkups. We ask them, hey, if you undergo this procedure, can some of that tissue go towards this research that we're doing? And then we move on that. So we end up processing quite a few samples because patients are very willing to see whether or not this kind of approach would work. Yes?
Given how quickly your lab has expanded, does this indicate that NIH is really looking on this as a positive kind of treatment option?
Oh, yes. Yes. I think that there's a lot of interest in this area, especially for figuring out, because everybody wants, it's not just finding a cancer early now, it's how do we deal with that cancer more effectively and how do we treat it? How can we make better drugs so that patients don't have to undergo this toxicity to drugs? So I think NIH is very interested. The lab continues to expand. And there's a lot of areas that we're kind of branching out into. We're starting to add more cell types to these organoid cultures. Something I haven't talked about yet is that I'm showing you guys the tumor, but what if we added additional components that comprise most cancer microenvironments, such as immune cells? Immune cells are a big part of what drives drug resistance in some cancers, especially things like pancreatic cancer. So what we're doing now is trying to incorporate more of these cell types and seeing how these organoid, if we can kind of automate and make this a more throughput method of doing this, and then assessing how these drug treatments are working in those cancers.
At some point you need to start to investigate autoimmune diseases too, I imagine.
Sure. Yeah, I think there's all sorts of directions we can go. It's really cool. Yeah?
So in your lab, do you actually try to help specific patients, like their doctors, figure out what to give them as a treatment based on how their cancer is growing in your lab or shrinking based on the treatments? Or are you just doing generic research to see if this is a viable technique for the future?
Mostly that.
Okay.
Now, we are part of a clinical trial that we just joined last year that's for rectal cancer patients, where we will be doing this as part of a larger study to assess more precise treatments for rectal cancer. I did show you there was one patient we did actually go through this process on, but in general, it's very much a early stage, making sure we can verify that this is an approach that can be used in a clinical setting.
does your approach effectiveness depends on the type of cancer? You mentioned mostly colorectal cancer.
Mm-hmm.
For other types of cancers, is the redox ratio is difficult to attain to an optimal level?
That's a great question. How does the cancer type actually affect some of these measurements? And the answer is that we can grow the one of the bigger challenges, really, because you can get the redox ratio from any of these cells. The harder part is growing some of these cancers into organoids that we can study, because it's dictated by how much sample you can get from a patient. So if a physician needs that for histology to assess whether or not that, for other parts of the study, we only get a small piece of that. And if we don't get enough of the tumor, then we may not be able to grow it into organoids. So it's typically more dependent on the front end of getting the necessary material to grow them into organoids than it is to actually do the imaging. Yes?
We just heard a talk about precision cancer treatments from a guy who's sequencing the tumor DNA. I'm having trouble putting together, what would be preferable, to know the tumor DNA or to know the redox ratios, or how do these two things, do we just not know enough about what chemotherapy works with what mutations, or what, I'm just having trouble integrating the two.
No, that's a great question. And so the question is, how does this imaging that I showed you guys, how does that work with something like doing genetic sequencing of a patient's cancer? And the answer is that I think the genetic sequencing is still front and center how a physician assesses what driver mutations are present. So if somebody has, say, a KRAS mutation in colorectal cancer, they know that certain drugs will be more effective for that type of mutation than others. Now, what the redox ratio can help decide is how effective that drug will actually be given these driver mutations. So I think they're actually, they're complementary, and you would want to do both, not just one, to assess whether or not a treatment would be effective.
Any other questions? One more?
Yeah.
There are some known complications in looking at organoids and what their behavior is when they're in the dish versus when they're in the person. And similarly, is there differences when you treat the thing in the dish versus in the patient?
Oh, that's a good question. Yeah. So is there a difference between, say, in vivo versus this ex vivo environment? And there are a lot of studies that are going on right now to basically look at the organoid in dish and then compare it to xenografts that you take the organoid and you actually put it into an animal like a zebrafish or a rat and study how they differ. And generally, the studies show that you basically get the same response in both cases. And I'm not going to say it's definitive at this point, but a lot of the more recent research has suggested that what you see in the dish is pretty reflective of what you end up seeing if you were to put that organoid back into a living animal.
Peter, before we go, Gabriella, would you like to stand up and say hi to everybody and let them know the meeting?
Yeah, so, hi, I'm Gabriella. I'm with the Morgridge Institute for Research, and Peter is one of our wonderful scientists, and I'm getting the Morgridge Institute for Research scientists out in the community more to tell you what we're doing. Morgridge is a private biomedical research institute that collaborates really, really deeply with the UW, and could not function without the UW. But because the resources Morgridge has, we can do a lot of interesting, basic, fundamental science research, and hopefully do it in very nimble ways that allows the scientists to take some really interesting paths. And I hope Peter's talk reflected the type of work that Morgridge does. So thanks.
Gabriella was the one that suggested Peter.
Yeah, thanks so much for coming.
Oh, thank you for giving me the suggestion.
Any other last ones? If not, thank you very much.